The seventh area of the regulatory science roadmap is to understand costs and performance trade-offs of NGS strategies in nuanced clinical contexts. What does this mean? In brief, it means that different regions of the genome will perform differently in an NGS test. Some regions will be easy to sequence accurately and precisely. Other regions will be difficult to sequence. And we know that there are clinically important genes in the easy-to-sequence regions, and there are clinically important genes in the difficult-to-sequence regions. So if you have a test that will be used for detecting variants in a difficult-to-sequence region, the performance requirements and potentially the test design will be different than a test used for detecting variants in an easy-to-sequence region. And of course, many tests will need to detect variants in both types of regions, particularly tests that use whole exome or whole genome sequencing. How do you design and validate an NGS test when analytical performance varies across the genome? Some examples of medically important genomic regions that are difficult to sequence include HLA and CY2D. In general, these regions of the genome may be difficult to sequence because of, for example, high GC content, the presence of repeats, or the existence of pseudogenes. As a result, these regions of the genome may produce lower coverage, reads that are difficult to align, and other problems that may lead to lower overall performance than that obtained with the same test on more well-behaved genomic regions. In reviewing an in vitro diagnostic test, FDA looks at the analytical performance of the test. For example, what is the accuracy and precision of the test? What is its limit of detection or sensitivity? For next-generation sequencing, this assessment may be challenging. Typically, FDA would evaluate each variant detected by a genetic test. However, an NGS test could potentially detect a variant at any position in an interrogated region, and there may be different types of variants. Even for a single gene, much less a whole exome or whole genome, the number of variants that may be detected can't be predicted or are too many to evaluate. For this reason, FDA has to infer the performance of the entire test based on performance on a representative set of variants. That set will depend on what the test is intended to detect. If it is intended to detect genes or variants in difficult-to-sequence regions, it will not be enough to demonstrate performance on easier-to-sequence regions, but rather using sequences with high GC content, repeats, or in regions with pseudogenes as applicable. When a test is intended to detect variants in a given genomic region, it is ideal to demonstrate sufficient performance of the test in that region. However, when performance is not sufficient, there are several potential options. The test could filter results from regions that lack the required performance. It may not always be desirable not to report those regions, and in some cases it might be possible to mitigate the risk of reporting them by including appropriate warnings and labeling or requiring orthogonal confirmation of results that don't meet the bar. In the end, however, further research is necessary. Here are listed some immediate needs. Reference materials that cover medically important regions of the genome need to be developed. We need better ways to improve test performance in difficult-to-sequence regions. Ways in which test performance over the whole genome can be inferred from a performance over a subset of the genome need to be developed. And of course, there are many other challenges that need to be addressed. By making it easier to develop tests with better performance across a wider range of the genome, these answers will also help FDA streamline its regulatory review of NGS. When addressing these questions, Precision FDA can be an important tool in the regulatory science toolbox. Because it is currently designed to assess and compare software performance, it can be used to identify new and improved informatics solutions to some of these questions. For instance, software that is developed or altered can be run using standard reference data files representing medically important genomic regions to assess comparative performance. And new reference sets can be developed and validated by running these through existing programs. In conclusion, research in this area of regulatory science is critical to providing new and improved strategies for the development, validation, and regulatory evaluation of NGS tests. This is an area that is important to everyone, not just FDA, and Precision FDA can play an important role in advancing this research. Thank you, and please contact me if you have any questions.